Chapter 1: Genetics. Now, have you ever wondered why organisms of a particular species do not look exactly the same? Or why children sometimes do not look exactly like their parents? The answer lies in the process of reproduction, which results in combination of hereditary factors that introduce differences between parents and offsprings. When organisms reproduce, the offsprings resemble their parents in terms of form, and this is because they inherit these characteristics from their parents. However, in other cases, the offsprings may show slight variations from their parents. The study of inheritance and variation is called genetics. In genetics, we study why organisms differ from one another in some aspects and at the same time show similarities in other aspects within a certain species. In this chapter, we will look at variation, and in the next chapter, we'll look at heredity. Variation refers to the observable differences amongst organisms, be it sex, height, color, complexion, and so on. It may be continuous or discontinuous. In continuous variation, there is no fixed value. Rather, there is a complete range of measurements from one extreme end to the other. Examples include height, foot size, body weight, skin color, head circumference, and so on. Continuous variation is often significantly affected by environmental factors. The environment where an organism lives has a significant influence on them. For instance, milk yield in a cow is not only determined by their genetic makeup, but also by environmental factors like food, weather, and so on. Continuous variation is graphically depicted by a histogram showing a typical bell-shaped distribution curve. In discontinuous variation, there are definite distinct group of individuals with no intermediate forms. Here, individuals fall into a number of classes or categories where features cannot be measured across a complete range. This variation is controlled by genes with little or no effect from the environment. You either have the characteristic or you don't. For example, you are either a tongue roller or not. You cannot be in between. It is the same with blood groups. You have one blood group or another. You cannot fall in between. Other examples include gender and handness. Causes of variations. Scientists now know that changes of various types happen in the genetic code itself, which increases genetic diversity among individuals and among species. These changes occur during gamete formation, fertilization, and also due to mutations. During gamete formation or the process of meiosis, variations in the genetic code can occur due to two events: crossing over and independent assortment. During process 1 of meiosis, homologous pairs of chromosomes lie side by side in a process called synapsis, forming a tetrad or bivalence. Once this structure has been formed, an event called crossing over can occur. During crossing over, a physical exchange of chromosome segments of non-sister chromatids occurs, increasing genetic diversity. In addition to crossing over, during metaphase 1, the pairs of homologous chromosomes align along the metaphase plate and sort independently into 4 daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the power of n. This means that an organism with 3 chromosomes can produce 8 unique gametes. For humans, where n is equal to 23, there are 2 to the power of 23 unique gametes, or over 1 million possible combinations. During fertilization, variation in genetic information is passed on to the offspring. You can think of the genetic information in a sperm cell and an egg cell as 2 bunches of play cards. A huge amount of genetic recombinations occur, a kind of reshuffling of the genetic deck. Mutations are spontaneous changes in the genetic makeup of an organism. For example, single-cell amoeba reproduce by copying their DNA, moving the copies to opposite sides of their body, splitting into 2, and going back into full size. When DNA is being copied, errors can occur which modify the DNA code. This is called DNA mutation, which is random and can occur anywhere on the DNA. It can introduce variation in the body form or function of the cell that receives the modified DNA. In this case, the modified cell has a longer arm. This cell will then pass on this change to its subsequent generations, and that introduces variation within the species population. And now we look at the DNA structure. Now, the structure of the DNA, or deoxyribonucleic acid, was fully described by Watson and Crick in 1953. It is a ladder-like twisted structure made of 2 strands joined by hydrogen bonds. Inside the cell, the DNA is double-stranded, in which 2 strands intertwine to form a double helix. Let's move to a more simplified representation of the DNA to discuss the details. We can unwind the double helix so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has 3 components: a 5-carbon sugar called ribose, a phosphate group, and one of the 4 nitrogenous bases: adenine, guanine, thymine, and cytosine. Nucleotides attach to each other by phosphodiester bonds. So the sugar and the phosphate groups make up the DNA backbone. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix. Although the nucleotides join through covalent bonds in the backbone, the 2 DNA strands interact through hydrogen bonds between the bases. Guanine always forms 3 hydrogen bonds with cytosine, while adenine always forms 2 hydrogen bonds with thymine. Each unit is called a base pair. This is called complementary base pairing and is crucial during DNA replication. Each strand of the DNA molecule is used as a template in the creation of a new double strand. DNA replication begins by the 2 DNA strands separating. Each strand is then used as a template for synthesis of a new daughter strand due to the complementary base pairing. The 2 double-stranded daughter DNA generated are identical to the parent DNA. DNA packaging. The DNA stores all the genetic information about a living organism in coded form. And as you can imagine, this information is enormous. In fact, a single cell nucleus has DNA with approximately 3 billion base pairs. If we store the entire DNA sequence of a single human cell in books, we would have 3,300 books, each with 1,000 pages, where each page has 1,000 letters. The length of the DNA strand in a single cell is approximately 2.2 meters. Now you can wonder how such a long molecule is packaged within a typical nucleus with a diameter of about 10 to power negative 6 meters or 10 nanometers. Let us learn about the packaging of the DNA. First, the DNA does not exist as one continuous long strand. Rather, it is split into several strands of varying lengths. Each strand is then wound around proteins called histones to form structures called nucleosomes, which is held in place by a protein called H1 histone. A typical nucleosome has around 200 base pairs. The nucleosomes appear as bead-on string structures. It's the nucleosomes that make up the repeating units in chromatin, the thread-like structures seen in the nucleus. These chromatin fibers coil further and condense to form short and thick bodies called chromosomes during cell division. In this way, the double helix DNA is packaged into chromosomes which are found in the nucleus. Next, we look at chromosomes, and we said these are the condensed form of the DNA. In a non-dividing cell or before interphase, the chromosome is made up of only one arm. After duplication during interphase, each chromosome has two arms called sister chromatids joined by a centromere. Sister chromatids have exactly identical DNA since they are copies of each other. Different organisms have different number of chromosomes. For example, humans have 23, dolphin has 44, platypus has 52, dove have 58, mosquitoes have 6, bees have 14, rice has 24, and so on and so on. And so you can see the chromosome number is not necessarily related to the complexity of the organisms. Within the body cells or somatic cells, chromosomes exist in pairs. Each pair has a characteristic length, and the members of each pair are called homologous chromosomes. Homologous chromosomes are similar, but not necessarily identical, as we saw in Form 3. Let's recall the human genome we had in Form 3. If you look at the human body cells, there are 46 chromosomes, and each chromosome has a homologous partner. This means there are 23 pairs of homologous chromosomes. In this case, the cell has not yet undergone DNA replication or the interphase. So we say that we have 46 non-replicated chromosomes, which is basically 23 non-replicated pairs of homologous chromosomes. Homologous pairs only exist in body cells, which are called somatic cells, and not in sex cells. Sex cells only have one member of each homologous pair, so they have half the number of chromosomes as the body cells. Homologous pairs of chromosomes have the same size, carry the same genes, and the genes occupy the same loci, except the X and Y chromosomes. They can have different alleles and are not held together by a centromere. During interphase, the cell undergoes DNA replication, and each chromosome now consists of two arms called sister chromatids held together by a centromere. Sister chromatids only exist after DNA replication. They are 100% identical, means they have the same size, carry the same genes in the same locus, and importantly, they have the same alleles for the same genes. After all, they are copies of each other. These sister chromatids are collectively known as replicated chromosomes. So now we have 46 replicated chromosomes, which is 23 replicated pairs of homologous chromosomes. And now we look at genes. Now, along the length of the DNA strands are sections called genes. The DNA sequences in the gene regions code for specific features of the organism, for example, a certain protein. And because DNA is packaged into chromosomes, then we can also say that a gene is a section of a chromosome. The position of a gene in a particular chromosome is called the gene locus. Again, considering that the DNA is packaged into several chromosomes, two genes may be located on the same or different chromosomes. DNA and protein synthesis. Now, the primary role of the DNA is protein synthesis. Each protein is coded for by a gene. In Form 1, we said that proteins are basically long chains of amino acids. Therefore, protein synthesis involves decoding the gene information into amino acids and then joining them to make proteins. This happens in the ribosomes. Protein synthesis involves two stages: transcription and translation. Transcription takes place in the nucleus, where the DNA acts as a template for synthesis of a messenger RNA or mRNA. It is governed by the principle of complementarity, with one exception: that adenine now forms a base pair with uracil instead of thymine. The mRNA is a single-stranded molecule that carries genetic information to the cytoplasm for protein synthesis, and hence the name messenger. The transcription process is different from DNA replication. In replication, the total DNA of an organism is replicated. However, in transcription, only one strand of the DNA gets copied into an mRNA. In case both strands act as templates in transcription, two types of proteins will be produced, one with the correct sequence and the other with a reversed sequence of amino acids. The part of the DNA that is involved in transcription is called a transcription unit, and it encodes for at least one gene. The section of the DNA first unwinds and the two strands separate. One strand is used as a template to synthesize the mRNA. Once done, the mRNA strand detaches. The mRNA then leaves the nucleus and enters the cytoplasm for the process of translation. The mRNA carries the coded information to the ribosomes, which will put together amino acids to make proteins. Another molecule called transfer RNA or tRNA carries amino acids to the ribosomes according to the coded information in the mRNA. But how does the ribosome establish the order of the amino acids? Well, the base sequence along the mRNA strand act as codes that spell out the sequence of amino acids. A set of 3 bases called codons represent a single amino acid. Each of the 20 amino acids have a codon or codons that represent them. This is called the genetic code. The genetic code is an array of 64 codons. Out of the 64, 3 codons UAA, UAG, and UGA are called stop codons, meaning they terminate the transcription process and so do not code for any amino acids. They are also called nonsense codons. The rest of the 61 codons code for the 20 amino acids. Notice that one codon codes for one amino acid. For instance, the codon UGG codes for amino acid tryptophan, and AUG codes for methionine and also acts as the start codon. However, several codons can code for the same amino acid. And so, the transfer RNA for each amino acid transfers the same to the ribosomes to add to the elongating sequence of amino acids. Once the ribosome encounters a stop codon, transcription is terminated and the protein is released from the ribosome. And now we look at mutations. We said that mutations are spontaneous changes in the genetic material of an organism. Mutations or errors that occur in the replication of the DNA can have significant impacts on individuals and on species. Mutations that occur within body or somatic cells can, for example, cause illnesses such as cancer if the mutation programs the cell and all its daughter cells to grow and divide uncontrollably. Mutations are broadly classified into two: gene mutations and chromosomal mutations. Gene mutations involve a change in the base sequence of a gene. These gene mutations can happen through substitution, insertion, or deletion. Occasionally, during replication, in spite of all the proofreading and corrections, a mismatched base pair in one of the chromosomes can happen. For example, adenine pairing with the cytosine base goes unnoticed and the cell proceeds with either mitosis or meiosis. Repair enzymes in the daughter cell that receives the mismatched base pair notice the mismatch and proceeds to correct it. However, these enzymes have no way of knowing which strand is the original parental strand or the newly replicated daughter strand. If the enzymes correct the mismatch by replacing the base on the daughter strand, the original base pair will be restored. However, if the enzymes correct the mismatch by replacing the base on the original strand, a base pair different from the original one will result, and a substitution mutation will have occurred. Insertions and deletions occur not due to any error due to replication, but rather from a break or breaks in one of the strands. Often, outside sources such as radiations or chemicals cause these breaks. Insertions often occur when one strand breaks in a single location and one or both of the free ends loop out. Repair enzymes fill in the resulting gap with new nucleotides, making the strand longer than the intact strand. After replication during cellular division, one daughter cell will receive a new larger segment of the DNA due to the insertion mutation. Deletion mutations occur if a strand of the DNA breaks in two places. The section between the breaks is forced out, and the repair enzymes draw the two ends together without filling in the missing nucleotides. Again, a loop occurs, but this time in the intact strand. After replication during cellular division, one daughter cell receives a shorter segment of the DNA due to the deletion mutation. Chromosomal mutations involve changes in the structure or number of chromosomes. There are 5 types of chromosomal mutations: deletion, duplication, translocation, inversion, and non-disjunction. Deletion occurs when a section of the chromosome breaks off and fails to reconnect and is completely lost. Deletion involves loss of genes and can have serious consequences on the individual. In duplication, a section of the chromosome may break off, duplicate itself, and the 2 pieces reattach to the same chromosome. This way, the chromosome length increases and results in increased number of genes. During inversion, a section of the chromosome breaks. It then rotates and reattaches itself to the same chromosome. This reverses the gene sequence and can have advantageous or disadvantageous consequences. Translocation occurs when a portion of a chromosome breaks off and attaches to another chromosome. Translocation can also occur when segments from 2 different or non-homologous chromosomes are exchanged. This leads to transfer of genes from one chromosome to another. This is called reciprocal translocation. Non-disjunction is a kind of chromosomal abnormality that results in addition or loss of one or more whole chromosomes. It occurs during the anaphase stages of meiosis. During anaphase 1, homologous chromosomes separate and move to the opposite poles. During anaphase 2, sister chromatids separate and again move to the opposite poles. Meiosis is responsible for production of 4 haploid cells from 1 diploid cell. Failure of the chromosomes to separate from each other during either rounds of meiosis results in non-disjunction. Normal chromosome segregation begins with the diploid cell at the start of meiosis. Let us focus on only one pair of homologous chromosomes. This pair can represent either sex chromosomes or any other chromosomes. Each chromosome consists of 2 sister chromatids joined at the centromere. During meiosis 1, homologous chromosomes separate, resulting in 2 cells, each consisting of 1 pair of sister chromatids. During meiosis 2, sister chromatids separate, resulting into 4 haploid cells. In animals, these haploid cells are called gametes. When they fuse with gametes of the opposite sex, normal diploid zygotes are produced. If the homologous chromosomes fail to separate during meiosis 1, then one cell is produced with both homologs and the other cell has no chromosomes. When the cells divide during meiosis 2, the sister chromatids separate and go into 2 of the resulting cells. These 2 cells are abnormal because they contain 2 copies of these chromosomes instead of 1. The other 2 cells have no copies of the chromosomes. Upon fusion with the normal haploid gamete, all the cells produced are abnormal for these chromosomes. Two zygotes will have 3 copies of chromosomes instead of the normal 2. These cells are said to be trisomic, and the condition is called trisomy. The other 2 zygotes will have 1 chromosome. They are said to be monosomic, and the condition is called monosomy. What if non-disjunction occurs during meiosis II? Homologous chromosomes separate normally during meiosis I, producing 2 cells, each having a pair of sister chromatids. If sister chromatids fail to separate in one of the cells during meiosis II, then 2 abnormal cells are produced, one having 2 copies of the chromosomes and the other having none. Normal chromosome segregation occurs in the other cell, giving rise to 2 normal haploid cells. When these cells fuse with normal haploid gametes, 2 of them produce the normal haploid zygote. The other 2 zygotes formed are abnormal, one being trisomic and the other being monosomic. In short, non-disjunction produces an abnormal number of chromosomes in a diploid cell. It may occur during meiosis I or meiosis II. Non-disjunction of the sex chromosomes is responsible for a variety of human disorders such as Turner syndrome, a female with only 1 X, and Klinefelter syndrome, a male with 2 X's and 1 Y. Non-disjunction of the autosomes is responsible for Down syndrome or trisomy 21, where there is an extra chromosome number 21.